Your Monstera is breathing right now. Every second you've been sitting there watching this, it's been making an active decision that could kill it. And you've never once looked at the mechanism actually making that decision. You've been told misting helps it breathe. You've been told a humidity dome helps it breathe. Neither one touches the actual system that decides whether this plant lives or dies. Respiration isn't a leaf function. It's happening in every living cell of this plant. In the roots, in total darkness, right now. Whether the grow lights are on or off. Photosynthesis and respiration are two separate systems, running at the same time. And if you've only ever thought about one of them, you've been managing this plant half-blind. By the end of this video, you'll know exactly how stomata are gated, why your root zone needs its own oxygen supply, and the specific setup mistake that quietly suffocates root respiration in some of the most expensive plants in this hobby. Start with what you think you know. Stomata. Little pores, they open, gas goes in and out. Simple. Here's the deception. Stomata aren't static holes. They're actively gated by a pair of guard cells shaped like two curved balloons, sealed together at both ends. When potassium ions pump into those guard cells, water follows by osmosis. The cells swell with turgor pressure, and because their inner wall is thicker than their outer wall, that swelling bows them apart. And the pore opens. This isn't passive plumbing. It's an ion-driven, moment-to-moment control system, responding to light intensity, internal CO2 concentration, and a stress hormone called abscisic acid that can force the gate shut in minutes. It causes those same ions to flood back out, draining the guard cells like deflating balloons and slamming the pore shut. Inside every one of those cells, two engines are running in parallel. Photosynthesis in the chloroplasts, converting light into sugar. Cellular respiration in the mitochondria, burning that sugar with oxygen through a multi-stage pathway. Glycolysis, then the citric acid cycle, then the electron transport chain, to generate ATP. That ATP is the actual currency the plant spends to build new cell walls, transport nutrients, and defend against pathogens. During the day, both engines run at once. At night, photosynthesis shuts down completely. Respiration never stops. Not for 1 second. Not for one cell. Now, here's the part almost nobody in this hobby has been told correctly. Roots respire too. Root cells need oxygen just as much as leaf cells, pulled in through root hairs from air pockets in the growing medium, then diffused inward, cell by cell, toward the vascular core. This is not a passive water straw. It's living tissue with its own constant ATP demand, running its own respiration engine 24 hours a day, entirely underground, entirely in the dark. But it gets worse. If those air pockets disappear, if the medium stays saturated, root respiration doesn't slow down politely. It starves. Oxygen can't diffuse through standing water anywhere near fast enough to keep up with demand. And a starving root cell doesn't wait patiently for oxygen to come back. You've seen what happens next if you watched our root rot breakdown. Without ATP, calcium signaling at the membrane breaks down. Without that signaling, the membrane itself loses structural integrity and starts to fail. Root death isn't caused by too much water. It's caused by suffocation. Respiration failure cascading directly into membrane collapse. The exact mechanism we're building on right now. Here's the bizarre paradox where collectors inadvertently sabotage their most expensive plants while trying to save them. You buy a sealed humidity dome to help the plant breathe better. You pack the surface with decorative moss to retain moisture. You water on a fixed schedule because a forum told you to. Every one of those habits attacks the exact system we just described. A sealed enclosure with poor airflow doesn't just trap humidity. It traps CO2 and depletes oxygen in the space right around the leaf surface. Guard cells respond to that shift by losing turgor and closing the pore. The opposite of what you were trying to achieve. Meanwhile, moss packed against the surface holds surface moisture against the medium below, collapsing the air pockets your roots depend on to respire. And here's a twist that breaks the more airflow always equals more breathing assumption completely. Not every plant runs the same schedule. Your Monstera runs standard C3 photosynthesis, stomata open by day. But Hoya carnosa, a completely different species, in a completely different plant family, runs CAM photosynthesis. Its stomata stay shut during the day to conserve water and only open at night, taking in CO2 and storing it as an acid until sunrise. If you apply one universal breathing routine across your whole collection, you are actively working against the biology of at least some of your plants. This is the trap. Symptoms of poor gas exchange: yellowing, soft growth, stalled roots. Get treated with more humidity, more misting, more sealing. Every fix reinforces the original problem, while the actual respiration bottleneck never gets addressed. It's a closed loop that costs money and never fixes the mechanism. So, if airflow, humidity, and watering schedules aren't the real lever, what actually is? Here's the protocol. Module 1. Restore the air pocket architecture. If you're growing in soil, this means a chunky, well-aerated mix, an aeroid blend with generous perlite or pumice, not a dense bagged potting soil. If you're in semi-hydro, Leca in a clear net pot does this by design, creating permanent air gaps around the roots that you can actually see. Module 2. Measure correctly for your setup. And this matters. If you're in Leca, do not rely on a capacitance moisture meter. Licea's air pockets are the whole point, and they read as false dry to a capacitance probe even with a full reservoir underneath. In semi-hydro, check the actual water line through your clear pot instead, and let the medium approach dry down between refills so the roots pull air, not just water. If you're growing in soil, that's where a capacitance-based meter earns its place. It reads actual root zone moisture in a uniform medium, so you're watering based on respiration needs, not a fixed calendar. Module 3. Airflow at the leaf surface, not humidity entrapment. Gentle, consistent air circulation prevents CO2 buildup around the stomata without sealing the plant off from gas exchange entirely. Crack the enclosure or skip the dome and run a small circulating fan instead. Module 4. Support the root zone biologically. Introducing beneficial microbes like Bacillus subtilis or mycorrhizal fungi to the root surface helps roots access oxygen and nutrients more efficiently, even in marginal conditions. This isn't a substitute for proper aeration. It's reinforcement once the architecture is already correct. Run all 4 modules together, and you're no longer fighting your plant's biology with guesswork. You're working with a respiration system that's been running successfully since stomata first appeared on land plants, roughly 400 million years ago. Your plant was never struggling to breathe because it lacked humidity. It was struggling because 2 respiration systems, one in the leaf, one in the root, were being managed by assumptions instead of mechanism. Fix the architecture, measure correctly for your setup, and stop treating every species like it runs the same clock. And if you want to see exactly what happens when that root respiration system fails completely, we already broke down the entire collapse cell by cell in our root rot investigation. Click the video right there on your screen, and I'll see you there. And if you want to see exactly what happens when that root respiration system fails completely, we already broke down the entire collapse cell by cell in our root rot investigation. Click the video right there on your screen, and I'll see you there.